We've got Dr. Moin Saleem joining us today. He is a professor of pediatric renal medicine from University of Bristol. Today, he's going to be delivering a talk on AAV gene therapy from basic science to clinical drug development in the kidney. It's going to be a translational science-focused talk, and we really want to stretch the audience here. So I will give the floor to Dr. Saleem now.
Okay, great. Thanks, Niall. Welcome, everybody, and well done, you guys, for organising this forum. My name is Moin Saleem. I'm a paediatric nephrologist. I'm a clinical nephrologist and do my clinical work at the Bristol Children's Hospital, and I also run a big glomerular disease research lab in Bristol. And for this talk, I'm going to tell you about one of our projects that we're moving into clinical development, which I think is really exciting, and I'm deliberately going to take you out of your comfort zone with some of the science, but hopefully you'll be able to follow the concepts, and also I'll start with some of the more basic stuff as well to get everybody up to speed. So I'm a nephrologist. I work on the kidney. The kidney, I sometimes call it the world's most sophisticated filter. Of course, it does much more than just filtering. But the filtering unit of the kidney is the glomerulus. And the glomerulus is a specialized capillary loop. I don't know whether you can see my arrow or not. Let me just make sure I've got a pointer. There we go. So it's a specialized capillary loop. And we each have about 1 million glomeruli. And it sits at the head of the nephron. So I hope most of you are familiar with the basic anatomy and physiology of the glomerulus. This is on the left a picture of an EM picture from looking from the outside onto the capillary loop with the podocytes on the outside. This is the capillary, and you can see these interdigitating foot processes that cover the capillary, and you can imagine that acting as a filter, as a filtering unit that stops protein getting through and allows huge amounts of water and small molecules to get into the urinary space here. And these are the podocytes that sit on the outside of that capillary loop. On the inside are endothelial cells, and this is the capillary space. And this is what it looks like in real life, another EM photo. And if you focus down onto that filtration barrier, so you've got blood on this side, and urine on this side. This is the basement membrane in between. These are cross sections of those podocyte foot processes, and on this side, on the blood side, you have endothelial cells. So this is essentially the filtration barrier, and the kind of nerve center, if you like, of the filtration barrier is here, which is the kind of cell-cell junction between adjacent foot processes. It's sometimes called the slit diaphragm, this little gap here, and that's what largely prevents the proteins from getting through into the urine. And what happens when that filter breaks down? Well, you get what's called nephrotic syndrome, which hopefully most of you have heard of. And here, what happens is essential plasma proteins leak massively into the urine, and that clearly causes large amounts of problems clinically. You can lose your antibodies, you can lose clotting factors, you lose albumin, which is the predominant circulating protein, and so on. And the clinical triad of nephrotic syndrome, the classic triad, is heavy proteinuria, hypoalbuminemia, low levels of measurable albumin, and then edema because of the osmotic changes between the blood and the tissue space. And so kidney disease is a huge global problem. If you get damage to the filtration barrier, you can gradually lose glomeruli through mechanisms such as fibrosis. That leads to chronic kidney disease, that leads to end-stage renal failure. And these are large public health problems, high healthcare costs. Clearly, dialysis and transplantation are very, very expensive. And it's estimated that the prevalence of CKD worldwide is about 8% of the population. So it's very common. And most end-stage renal disease originates in the glomerulus. So that ranges from the kind of commoner conditions such as diabetic nephropathy, aging. As we age, we lose glomeruli, hypertension, etc., to rarer conditions, and some of these are monogenic conditions, so genetic conditions, congenital nephrotic syndrome, which I'll talk about, Alport syndrome you may have heard of, and then various immunological and inflammatory diseases that are sitting here in the middle. So congenital nephrotic syndromes are due to mutations in genes, and the genes that are mutated are in the podocyte, so that they're expressed in the podocyte. So this baby had a mutation in the nephrin gene, NPHS1, and that the protein nephrin is only expressed in podocytes. So if you get damage to the podocyte through a genetic mutation, you get a massive leakage of protein, and you get these nephrotic syndromes presenting very early in life. These are rare. As I say, they're inherited because they're genetic, so usually autosomal recessive. You get early onset of proteinuria, usually a rapid onset of renal failure, because these are untreatable conditions. They don't respond to steroids or any other agents, and the only way of managing them is with transplantation. So this is a breakdown of the glomerular filtration barrier at the podocyte level. And there are lots of genes associated with nephrotic syndrome. There's currently at least 80. So this isn't a comprehensive list, but just to illustrate that some of them are associated with syndromes. So it doesn't just involve the kidney, but you can, if you get mutations in these genes, you can get problems with other systems as well, such as Alport's disease, which affects also the ear and the eye, for example. And then there are lots that are associated just with renal disease or just with podocyte disease. So again, just to illustrate this, NPHS1 is the one I just mentioned, which is in the protein called nephrin. And NPHS2 is the commonest of all of them. It's about 20% of all patients. And the protein that it codes for is called Paedocin. And these are remember those two, because those are two that I'm going to talk about as we move along. And these genes express their proteins in the podocyte. And so as they've been discovered over the last 10 to 15 years, it's taught us lots about the biology of the podocyte and what's important in terms of which proteins are required for intact filtration. And there's lots of proteins at the slit diaphragm that I mentioned that are important. There are also proteins that are important in the mitochondria. So mitochondrial disease proteins can cause nephrotic disease, and other areas such as transcription factors, which are genes that affect that are expressed in the nucleus and affect expression of other genes. And that's led to changes in clinical practice. So because we've discovered genes that can cause nephrotic syndrome, when we now diagnose a patient, a child with a nephrotic syndrome, we do a gene testing panel, and that tests for all the known genes and has a relatively rapid turnaround. So it's affected our clinical management protocols. So the question then for us as researchers is gene replacement therapy an option? So can we replace the faulty genes with genes that with the normal gene that works normally? And this is called gene therapy. And gene therapy for human disease has been postulated for quite a long time. So this is over 50 years ago now. It's been hypothesized that human genetic disease can be treated with gene replacement, but clearly at that time the technicalities were pretty much unknown, and so there were both scientific and ethical problems. And so the questions are, how do you get your DNA, your correct DNA, to the correct cell? How do you stop it then being broken down? How do you ensure it's targeted to the right place, to the nucleus, so it harnesses the cell's nuclear machinery for transcription? And then you've introduced a foreign protein. How do you prevent the immune system from attacking it? And so can we ensure that this whole process is done without toxicity or side effects? And so gene therapy started probably in the early 90s. And there were two main routes that people tried. The first was called ex vivo, and the second was in vivo. And so with ex vivo gene therapy, you take the cells out of the body here. You expand them in culture. So this is really for circulating blood cells, lymphocytes, etc. You expand them in culture. You use a vector, a viral vector. I'll talk a bit more about that. To introduce your genetic cargo into the cells, and then you put the cells back into the patient. So that's ex vivo, and then in vivo, you can directly inject your viral vector into the patient and hope that it goes to the right place. And that's one of the key technical challenges of getting it to the right place. So the first trial in the world was in 1990. This was a patient diagnosed very early in life with a severe combined immunodeficiency, so an enzyme deficiency that caused immunodeficiency, and he was given an enzyme replacement therapy. He was given extrinsic enzyme replacements, but that wasn't working. So he had a retroviral mediated transfer of the actual gene into his mature T cells ex vivo. That normalized his number of T cells, gave him a normal lifestyle, and he's still alive today, so highly successful. However, there were problems. So that then led to more trials. There were two main trials running in Paris and in London that did the same kind of thing, stably corrected immune function in T cells, but 5 out of 20 patients developed leukemia. And this was because the vector, which was a viral vector, gamma retroviral vector, integrated near oncogenes, which caused leukemias. And one of those patients died. And then there was a direct in vivo approach that was tried using adenovirus this time as the viral vector. So these viruses are used effectively as Trojan horses to take your gene cargo into the cell. And this was for another enzyme deficiency, directly into the hepatic artery. This was an 18-year-old boy, but he developed very severe early rejection of this virus. He developed a severe immune reaction, diffuse vascular coagulation, and multi-organ failure, and sadly he died. So in the beginning of this century, gene therapy really wasn't doing very well after a promising start. And some of the lessons learned was that adenovirus, which was used as the Trojan horse vector, very immunogenic. It's good for cancer and vaccines, as we know, for COVID and so on, but not good for gene therapy. And so we needed to kind of refine these retroviruses and other types of viruses to be more acceptable, particularly in vivo. The ex vivo retroviruses are actually still working pretty well and with good long-term expression. So along at that time came adeno-associated virus. This is a small virus, 20 nanometer single-stranded DNA. It's not pathogenic, so we're all exposed to it but don't get ill. And it's episomal. It doesn't integrate, so it stably sticks around the nucleus but doesn't integrate into the genome. So it doesn't have that cancer risk, has relatively low immunogenicity, but one of the drawbacks is that it has what's called a small packaging capacity. So you can only put a small amount of your cargo DNA in. So if your gene is large, then that becomes a problem. So and the way that it's done is that you take the genetic sequence of this of the virus, you remove the replication and the capsid elements of the gene, and you replace them with your transgene, so they're your gene of interest, so the virus can't replicate once it's in there because you've removed that. The capsid is introduced separately, so that's the capsule of the virus. And so up till now, then, since that technology has been introduced, there's still good ex vivo gene therapy using these previous viruses I talked about, but there have been clinical trials that show efficacy in vivo now of viral gene therapy in monogenic diseases using AAV, including in the eye, in the liver, and in neurons, and these are some high-profile successes in these diseases. And this is just showing some of the successes. So hemophilia, you all have heard about. So that this is AAV delivered the hemophilia gene to the liver, retinal gene therapy, and spinal muscular atrophy, which also has been in the press quite a lot, very successful in this very severe disease. So far, we've got successes in monogenic disease in other organs using adeno-associated virus. The question we asked ourselves is, can we do the same for a monogenic kidney disease? And the question was, in all of these successes, where is the kidney? There's been no successes so far in the kidney space. And that's partly down to the fact that the kidney is so complex. So it has lots of highly specialized cells, at least 40 different highly specialized cells. And so targeting the right cell type and without having off-target effects elsewhere has proved really quite. So those are some of the main hurdles, as I say, targeting the correct cell type, the cargo capacity I've mentioned. The gene has to be of a suitable size. Will it stay there long enough? If cells turn over, then you lose because it's not integrated, you lose that cargo. Can you get the right dose, minimize the dose, and minimize off-target toxicity? And so some of the key approaches that are used for specificity, and this really is the key to gene therapy, is getting your virus to the right cells. And so these are some of the approaches that we've now used to try and get specificity into the kidney. Firstly, you can use cell-specific promoters. So as I mentioned before, nephrin was a protein that is mutated in congenital nephrotic syndrome, and nephrin is only expressed in the podocyte. So its promoter is specific for podocytes. So that's very useful. We can use that. And then the capsid of the AAV. AAVs have lots of different capsids, and the different AAVs have different tropism for different cells and also in different species. So that needs to be tested. You need to test one that will work for the cell type that you're interested in. Can we achieve local delivery to the kidney? And I'll give you some data on that. And will the gene of interest be expressed long term in the cell once you get it there? Because clearly you don't want to be, you can't keep redosing the patient. So the key question was, can we use some of these gene therapy approaches to target the podocyte? As I've said, the podocyte is the key cell within the glomerulus, and the glomerulus is the key then to most kidney diseases. So these are some of the different ways you can get nephrotic syndrome, some different insults, if you like. And some types of nephrotic syndrome are immune mediated, and these are so-called steroid sensitive, often steroid sensitive patients, and they're thought to be caused by immune mediated circulating factors. A proportion of nephrotic syndromes are steroid resistant. Some of these are still caused by the immune mechanisms, but a large proportion, and in children it's at least a third of these patients, are caused by single gene mutations. And there are no effective medical therapies for these. And the most common, again, I've said this already, the most common is the gene affecting NPHS2, which encodes a protein called Paedocin. And this is another slip diaphragm protein. So it's only expressed in the podocyte and only expressed at the slip diaphragm. And this is the sort of clinical phenotype you get in these patients. It's severe, it's progressive, it usually presents in childhood, usually early childhood. Most patients will progress to renal failure within 4 years, and there's no therapy apart from dialysis and transplantation. And the underlying cause is mutations in these podocyte expressed genes. As I've already mentioned, there's over 80 of these, and Paedocin is the most common cause. These are still rare. So in the US or in the EU, there are under 200 new cases per year, but there's still a significant problem, particularly in pediatric nephrology practice. So bringing you back to the mechanisms that or the techniques that we want to use. So we want to use adeno-associated virus to introduce our transgene, and the transgene in nephrotic syndrome with the Paedocin mutation is NPHS2. So we want to put NPHS2 into there and introduce that into podocytes. So this was our aim. And the advantages of targeting the podocyte are, you can argue that it's almost a sort of perfect cell for gene therapy, because firstly, it's a terminally differentiated cell. So a bit like neurons, once you've got your podocytes, you've got them for life. You've got the same podocyte for life. So it doesn't turn over. Therefore, if you can get your gene cargo into that cell, it should stay long term. And as I've said, we have podocyte-specific promoters, so we can directly, so even if you put your virus into lots of different cells, the promoter, if you create your construct correctly, the promoter will only allow the protein to be expressed in the podocyte. So that was the plan. Now we get down to some technical sort of details, and I'll try and sort of take you through this step by step. But firstly, you've got to make your construct. So as I said, this is the kind of viral genome. ITRs are the flanking regions of the meat of the genetic material, if you like, and this is the genetic material that makes the proteins for the virus. And what you do is you take out the capsid and the replication machinery, and you replace it with the correct promoter, which I've talked about. These are various cassettes that help with translation of RNA into protein, and then you can put your genetic cargo of interest. So either we've used a green fluorescent protein, so that shows that glows green, so you can see in the cell whether you've got your protein expressed, or you put your the actual gene that you're interested in. So Paedocin is what we've used, or mouse Paedocin in some of the mouse experiments. And we also use specific capsids. So there was a trial and error stage of trying different viral capsids to see which ones transduce our cells best. And we came up with these ones for human cells and this one for mouse cells. So that bit, I'll just sort of show a bit of data on how we did that. So we tried various different AAV capsid serotypes, both in mouse and human immortalized podocytes. So these are podocytes that you can grow in a dish. Without going into the details, what we can see is in the humans that LKO3 was the best capsid with the best, these are fluorescent, so you can see the fluorescent protein in the cells. And in mice, it was a different serotype, which was called AAV9. So we knew which capsid we wanted to use. We then wanted to test whether, once we put the virus with our protein into the cells in a dish, do they express that protein? So for this, we used GFP, which is a fluorescent protein. Again, there's lots of data here. I'll just show you visually how it looked. So this was human podocytes, or human endothelial cells, or human proximal tubular cells. So we're testing which ones go into the podocytes best. And as you can see top left here, the fluorescence is brightest here. These are two different promoters. So this is CMV, which is a generic promoter. This is the NPHS1 nephrin promoter, which should only go into podocytes. So that's the key panel here compared to this one and compared to this one. You can see that there's nice specificity for podocytes using that podocyte-specific promoter. Then we've got our testing of viral transduction of the actual gene of interest. Sorry, just trying to get this little somebody in the room. Because viral construct that we've got, and then we again transduce that into the podocyte. This time it's got a tag HA, so the HA we can show up green again, and you can get nicely in the podocytes here. These are the podocytes which are like the protein of interest.
Hi, Dr. Saleem, sorry to interrupt. I think you've just cut out there. Would you be able to start
and then
the slide from the beginning?
Okay, can you hear me?
It's just cut out a bit. Would you be able to start explaining the slide just from the beginning? Because I feel like you cut out for about 20 seconds there.
Okay, but you can hear me now, can you?
It should. I think it's okay now. Yeah.
Yeah. Okay. I'll start this slide again. So, now we're doing testing of the actual gene that we want to put into the podocytes, which is the Paedocin gene. So this is the viral cargo. This time with the Paedocin gene as our gene of interest. Here we are putting the virus with the gene into human podocytes in a dish. These podocytes are deficient in Paedocin because they've got the mutation, the commonest human mutation. So they're derived from a patient with a mutation. So they don't express Paedocin normally. In green, you can see what we've attached is a tag, an HA tag, so that glows green on the cells. So you can see that that's expressed nicely in this cell here. What we want to also to see is once you've expressed your your protein of interest, does that rescue the phenotype of the cell? And these cells, when they're deficient in Purespring, they have what's called an adhesion defect, so they don't stick to the plastic in the tissue culture well very well, and you can measure that. So they have low adhesion, which is shown here compared to normal, and then when you add the Purespring back using the AAV virus, that adhesion is rescued almost back to normal levels. So you can show both protein expression and functional rescue. So that's all cells in a dish. We now want to see what happens in a living animal. So we can do a mouse experiment for this. So what we did is we took a mouse and we can knock out Purespring in its podocytes. So this is called an inducible knockout of Purespring. So construct in the mouse, and I won't go into the details, but it allows you to knock out Purespring by giving it tetracycline. So it's a chemical stimulus will knock out the Purespring in the podocyte, and that therefore gives it nephrotic syndrome at a time that you can manipulate. So what we did is that we gave the virus to the mouse. So it's an intravenous injection to the mouse where we give it the virus with normal Purespring, with a nephrin promoter, so it should only express in podocytes. And then 2 weeks later, we induce the disease in the mouse by giving it tetracycline, so it gets nephrotic syndrome. And then we follow that mouse for the next 16 weeks in terms of measuring proteinuria. And at the end of the experiment, looking at the blood and kidney profiles. And this is what happens. So these mice, the black is the control mice, so they don't have the virus, they don't have the rescue. And this is proteinuria, so they gradually develop massive amounts of proteinuria over a period of time. And the treated mice, this is 2 different promoters we tried, but they both worked equally well, show much lower degrees of proteinuria as time goes on. And that correlates with nice histological improvement. Again, I won't go through all these in detail, but you can see it visually best down at the bottom here on electron microscopy, where the untreated mice have a destroyed filtration barrier. The treated mice have these nice podocyte foot processes. Actually, I've lost my pointer, haven't I? Let me get that again. The treated mice have these nice foot processes that you can still see compared to this kind of destroyed filtration barrier. And you can measure the histology, you can quantitatively measure the histological parameters. So you got a nice rescue of histology. And I think this is probably most impressive is long-term survival. So these mice, when you induce disease, they don't survive more than about 100 days. So they die of very severe renal failure relatively early. However, if you treat them, this was our pilot experiment, about 2/3 of the mice survive beyond a year. So pretty much the lifespan of a mouse. So that shows that you can achieve really good long-term survival with this therapy. And then we sort of analyzed, we wanted to ask the question, how many of our podocytes are expressing the gene or expressing Purespring in this model? And what you can see here, easiest seen here. So the green is a tag for the podocytes, and the red is where we've got that HA tag where you can see our expressed protein from the virus. And so you can see them scattered around the red. When they're co-localized, it looks yellow. So we quantified this as roughly 10 to 20% of podocytes were expressing our gene of interest. And even with that 10 to 20%, you were getting pretty good rescue of function and lifespan, which was very encouraging. That experiment was rescuing the disease, was giving the virus before you induce disease, which clearly in clinical practice is not what you can do. So we wanted to do an experiment where you induce the disease first and then rescue the phenotype once you've got proteinuria. So this was a slightly different model. It's called a knock-in model, where we put the specific mutation, the commonest mutation that you get in man, into a mouse model. and we induce disease again. So we induce disease, about 2 weeks later they get disease increases to a very high level. The decreases is and the shuts down. And then when it's the green line about this, you can see that the proteinuria by the end of the experiment had come back down.
Hi, Professor Saleem. You've been cutting out again for about the past 15, 20 seconds.
And you can see that extra and the previous.
Yeah, if you could start from the beginning of the slide, that'd be great. Yes, a little bit better.
If I disappear completely. Yeah, okay. I don't know why it's cutting. Yeah, I've got. Okay. Okay, I'll start this line. So, I've shown in the previous introduced are Paedocin, and the yellow is the co-localization of the two. So we're showing pretty robust expression of Paedocin. And then the histology here, again, you can't see it very well, but you've got pretty much normal histology in the treated mice compared to, if you can see the sclerosed glomerulus here, which is what you get if you don't treat them. So this is a nice proof of concept that you can successfully target the podocyte with a specific promoter. That leads to Paedocin expression, and that leads to long-term rescue of a very severe disease. The other thing you want to that's really important in these experiments, because this is going to be important if you take this into clinic, is what's the off-target expression, and could this be causing potential damage or problems elsewhere? So we looked at all the other organs in the body, and this is in the mouse, and we saw some expression in the liver, some expression in the spleen of the virus, and this was probably because this is an intravenous dose, so it goes around all the organs, and it's a large dose. So we need to get around that problem. We're going to get safely into humans. So the next stage is that you want to make that into a drug development program, and this is where the sort of commercialization comes in, and here you have to raise money to do all the kind of correct stages of drug development, which includes further experiments, toxicity studies, and all the kind of regulatory hurdles that you have to get through before you're allowed to put a new drug into a human. So we formed Purespring Therapeutics with an investment from a venture capital company called Syncona. These are investors who've invested in other gene therapy companies. So they gave us an initial funding of around $60 million. This was about 3.5 years ago. That enables us to build the team with the experience to take these kind of preclinical results through to first-in-man trials. And this includes things like manufacturing. So there's, again, I won't go through all the details, but you need clinical grade production of all the different components that I've talked about, the plasmid DNAs, the cell lines, and you need manufacturing facilities that can deliver the amounts and the quality that you need. And then you need to do toxicology, so large animal testing of a clinical grade product to look at off-target effects and whether we can achieve direct renal delivery instead of that systemic delivery that we did in the mice. So this is really important to smooth that path to clinical utility, and it takes time and it takes a large amount of money, as you can see. So in terms of that direct delivery, this was a key part of what we wanted to achieve, because as I've shown you in the mouse, you get quite a lot of off-target delivery of the virus, and that could be a problem, and that might not allow you to get through the regulators like the FDA to allow you to go into human trials. So we needed a novel administration method that delivers locally to the kidney, and that's what we've worked on over the last couple of years. And the advantage of that is you can optimize the efficacy of what you're delivering with lower doses, you limit the systemic exposure, and it also improves manufacturability. So-called cost of goods is lower because you're using lower doses. And this is the data that we obtained now in a pig. So we're using a large animal model. Interestingly and importantly, it's exactly the same size as a small child that you would dose. So that's helpful because the size of the kidney and the size of the anatomy is exactly the same. So we can directly test the methods that you would use in the clinic. This is gene expression in the glomeruli. So you can see on the low power all the different glomeruli here. The purple is showing you the expression of the gene that we got using a direct local delivery. And this is a high power of a glomerulus, and you can see that there's lots and lots of purple there. So you're getting high amounts of gene expression. This is now at the mRNA level rather than the protein level. And this is just showing sort of several pigs and the amount compared to untransduced controls. And you can translate that into this is what I showed you with the mice. You can see that the Paediatric that we've introduced, so the nephrin here is marking the podocytes. The Paediatric is what we've introduced, and you can see it's pretty much 100% here. So it's going into all of the podocytes. It's far more efficient than we ever saw in the mouse. Just to show you the direct comparison again, but at the mRNA level between mouse and pig, this is what we saw in the mouse, this is what we see in the pig. So we're seeing, as you can see visually, much higher levels of expression in the pig using that direct approach. And this is about 10 times lower dosage than we used here in the mouse. So at much lower dosage, we're getting much better expression. So that's really positive. And then looking at the biodistribution in other organs. So this is a treated kidney, and this is now looking at the vector genomes. So this is just looking at the virus, how much virus has got into each of these organs. This is the treated kidney. This is the untreated kidney. So obviously, when you directly inject into one kidney, the other kidney is left alone. So you get much less. This is in the liver. The liver is usually a sink for any virus that you put into the body, and you've got very low levels in the liver. And as I say, this is how much virus gets in. There is actually no detectable protein in the liver seen, so that's really encouraging, and in the spleen even less. So that gives you lots of advantages in terms of dose, in terms of manufacturing your drug. At the end of the day, you can use much lower doses locally, so that's the doses, how it's measured, compared to what's used systemically in other gene therapy trials that don't have that local delivery advantage. Because the kidney weighs much less, you can make far more doses per batch of manufactured virus when it comes to making a commercial batch. So we're now in a very strong position to start to clinical trials in different glomerular diseases, because we know that we can directly target the podocyte. We know we can limit the off-target effects, and we know we can use a low dose. And then you can therefore use a sort of plug and play approach. Most of these components will be similar for various different glomerular or podocyte diseases. So the so-called helper and capsid plasmids are the same. The plasmid, the DNA, which carries the therapeutic gene, is the plug and play bit, and all the steps towards manufacture are then exactly the same by the time you've got to a drug product that you can put into humans. Again, the promoter is always going to be the same. So that allows you to think about various different diseases that can be treated via targeting of the podocyte. And so coming back then to some of the sort of clinical implications here, the podocyte is important for lots of different aspects and biology of glomerular function. And when it goes wrong, that includes things like maintenance of the architecture, for example, by production of collagen in the basement membrane. It regulates filtration through those slit diaphragm proteins. It actually regulates the immune environment within the glomerulus. So we know that the podocyte produces lots of complement regulators, for example, and can remove immune deposits. And it has a key insulin sensing response. And this is really important because it's one of the few cells in the body that's directly insulin sensitive. And that could be absolutely crucial in the development of diabetic nephropathy, which is the commonest cause of renal failure worldwide. And so some of the diseases that are related to those functions, so basement membrane all port syndrome, the slit diaphragm proteins, these inherited nephrotic syndromes that I've talked about already, the complement regulation, there are lots of inflammatory diseases in the glomerulus like membranous nephropathy, IgA nephropathy, and insulin sensing is diabetic nephropathy. So they're both non-monogenic and monogenic disorders. Targeted if you can put in the correct genes that you think will reverse the pathways that are dysregulated in any of these diseases. So just sort of a little aside about newer delivery methods that people are working on apart from AAV. There are things called lipid nanoparticles. So these are non-viral vectors where you have a lipid capsule that carries your payload. And they can carry larger payloads. They're inert. You can redose because you don't get immunogenicity. But the targeting is really difficult. It doesn't lead to permanent gene expression, so you will need to have redosing. And the efficacy at the moment is pretty poor, to be honest, of these types of technologies. And then there are other viruses. In fact, baculoviruses are viruses that infect insect cells. And they've been modified to infect mammalian cells by various different scientific groups. And they have an enormous cargo carrying capacity. They could probably carry a whole chromosome. They're non-pathogenic to humans, but they're not yet approved for human clinical trials. And then there's gene editing. You'll all have sort of come across gene editing in various forms, CRISPR-Cas technology. This is relying on double-strand breaks in DNA at specific locations using these CRISPR-Cas enzyme and targeting mechanisms. So you have a guide RNA that directs this Cas9 protein to the target, which cuts it, but it can lead to off-target effects and unintended mutations. So that's not yet, or it's just starting to sort of be used in the clinic. There's one very recently approved for sickle cell disease. Prime editing is a more up-to-date version of the CRISPR-Cas technology that doesn't require double-strand breaks, so it reduces that risk of off-target effects and uses these things called pegylated RNAs. So again, I won't go into the detail, but they are much more specific and again could be used in the future. So just to sort of finish off then, I hope that I've shown you some of these key concepts. Podocyte looks like an ideal target for targeted AAV gene therapy with specific promoters, long-term gene expression. Gene delivery to the podocyte is practical in vitro and in vivo, and we've shown rescue of genetic nephrotic syndrome. Local delivery is also possible, and we've shown that works really nicely. Limits toxicity, limits the dose requirement, improves efficacy. So that opens up the whole field of gene therapy for glomerular disease, both monogenic and non-monogenic. So it really could be transformative for glomerular diseases. And we're envisaging the first clinical trials within a year, and hopefully even before the end of this year. So that's the current plan. So I'll finish there, and I'll hopefully. I've been asked to share with you the QR code for the chat, but hopefully you'll get some questions.
Thank you very much, Professor Saleem, and thank you all for joining us today. I hope the session's been informative. I hope it's been valuable to you. Please don't hesitate to put any questions into the chat, and Professor Saleem will be happy to answer them if you can just write them in the chat. And also don't forget to scan the QR code.
And it doesn't matter how stupid the question, because as I say, I've deliberately tried to push the boat out and put you out of your comfort zones.
Well, just to give everyone some time to think, is it fine if I start by asking you a question, Professor?
Yeah.
I wanted to go back to one of the slides you had at the start, and you were talking about how patients were rejecting, patients with severe combined immunodeficiency were rejecting some of the retroviral vectors. I just found that a bit confusing, because I thought if someone's immunodeficient, how can they reject a viral vector?
That was actually in an enzyme deficiency that wasn't immunodeficiency. It's like a metabolic disease in that particular patient. So I should have explained that possibly a bit clearer. But the patient who got this sort of severe immune reaction was I think it was an OTC deficiency. So it's a metabolic disease rather than immunological disease.
Okay, thank you. If anyone else has any other questions, you can write them in the chat. Otherwise, if there's no questions, then we can wrap it up. Oh yeah, we have a question.
Okay, so why is the AB chosen as the most practical viral vector to use? So there are lots of different viruses, as we all know. We're all familiar with viruses since COVID, and they can infect. They're all very efficient at infecting cells, so they'll all do that very well and take genetic material into a cell. But the problem is that you don't want the virus to damage the cell. You don't want an immune reaction to the virus once you've introduced it. Also, many viruses take their genetic material and integrate it into your own genome, so that can be really dangerous. So AAV precludes most of those problems. It doesn't integrate into the genome. It's epigen. It sits around in the nucleus. It's so-called epigenomic, it's non-pathogenic, so it doesn't harm your cells, and it has a relatively low immunogenicity, so your own immune system doesn't tend to react to it. So that's the reason that it ticks most of the boxes for being a successful virus for this. There's a question about the research funding. Yeah, no, it's a good question. We all academics worry about funding. So once you've got to the stage of wanting to develop a successful sort of clinical academic project into a therapy, that's where government funding, there's a so-called translational gap. There isn't enough funding from government grant agencies, because you need a large amount of money to do it. Most government grants will sort of top out at maybe 2 or 3 million, and around that kind of amount. In order to start this, in order to get to a clinical product, that takes at least about, we've so far have investment of just over 100 million from our first and second rounds of funding. So you need to get external investors, and that's why we use venture capital. Sometimes pharma companies come in, big pharma companies, and invest in these, and they're the ones that ultimately will get most of the profit out of this if it becomes profitable. So that unfortunate consequence of capitalism, but that's the way it goes. But that's why we had to start a company based on external funding rather than government funding. It's very unusual to be able to get government funding to take you all the way through to a clinical product. It does happen sometimes, particularly for some very rare diseases, but it's pretty unusual.
Okay. Right, well, if there's no more questions, then I think it's okay for us to wrap this up. Before you all leave, I will post a feedback form in the chat. We'd be very grateful if you could fill that out. And thank you very much, everyone, for joining us today. I hope you found it informative. And thank you very much, Professor Saleem, for delivering this talk. We all really appreciate it.
Okay, pleasure. Okay, good night, everyone.
Thank you. Bye-bye.
Bye.